vaccines by the Department of Biotechnology, Stella College, with the support and warm wishes from our principal, Dr. Sister Jacinta Quadras. The rapid spread of SARS-CoV-19 has led experts to declare that it would continue to spread if a vaccine cannot be found. Vaccine development is typically a long game. Cases have topped the 7 million mark on June 8, 2020 globally. India too is seeing a surge in the number of cases every day. While there's still a long way to go before we put the pandemic behind us, trials and research are going on to rap speed to attend to the crisis at large. Many potential treatments have shown promising results in the clinical trial stage. While some are carrying on production, we recap all the recent development as the world waits for a COVID-19 vaccine with bated breath. Today, Today, we have an eloquent speaker, Dr. Rajkumar Utupuri from University of Frankfurt, Germany, to share the pipelines of vaccine preparations about COVID-19. Now, I request Ms. T. Rajeshwari from the Department of Biotechnology to introduce the speaker to all the participants.
Vijayawada from Stella College introduced Good afternoon to one college. Today I'm honorable speaker Utupuri Garu, fellow at University. His list of achievements provide a special mention achieved accolades at a very young age. A degree from University, a master's degree in University. There is one Bharatidasan University, Jason. Academic record in his bachelor's, his PhD thesis of sphingolipid mechanism underlying LV. Dr. Rajkumar Utupuri Garu, like graduate school, Goit University, assistant from he has so many publications three diseases, state of angiopoietic brain barrier size are PTP dependent PIE2 signaling. Blockade, amino A and alternation metabolism in an stroke. How many member of Neuroscience Society.
ప్రార్థన చేస్తే మనకి
Indian Society
జరగదు.
applied
నేను నా పర్సనల్ ఎక్స్పీరియన్స్ కూడా
member of
కొంచెం సిగ్గుంది ప్రార్థన అడక్కే చెడతానమ్మా
2020
అయిపోతే ఏముందిలే
of the chapter on
మనం నెగ్లెక్ట్ చేసి లేకపోతే ఆ టైంకి ఏదో ఒకటి
also a publisher. He BioBricks conferences on bioindustry, IT and JM, not least, Buturigalu is a presented under 12 Krishna ANC softball and represented Krishna Hyderabad. He's BMW Marathon 2013 Germany during 2000. I invite Dr. Rajkumar for the session.
Hello. Yes. Actually, the voice is little breaking, but I hope my voice is clear for all of you.
Yes, sir. Your voice is clear, sir. You can go ahead, sir.
Okay.
Sir, one small instruction, sir. Sir,
Yes.
please click on presentation, sir, when you are presenting.
Yep.
In between, if any of our participants click on presentation and interrupt your screen, just ignore them and click on your presentation, sir.
Okay. Okay.
So that we can avoid the confusion, sir.
Yeah. Yeah. Okay.
Disturbance also we can be avoided. Yes, sir. Thank you and go ahead, sir.
But please, whoever are participating, please be a little silent or mute on your audio, because previously I could hear a lot of voices who was speaking. So I please request about that. Okay. So thanks a lot for all the introductions and respected faculty members, whoever is participating, and my dear students and everyone who is joining. Warm greetings, whoever and wherever they are in different time zones, and hope everybody is staying very healthy. And it's my pleasure to address you all and share with what I know about the trends in the vaccines and what are at what developmental stages are about the coronavirus, and just will give you my expectations on when could be the end of the pandemic. And what I'll do in my presentation is, because I think the world is full of the information on corona, and I don't want to talk too much about very basics of this. And I'll try to give you a brief introduction about some aspects, and then I'll be taking you through what is the mechanism, how the SARS-CoV is infecting, and then I'll start discussing about the COVID-19 drugs and what are the vaccines in trail, and maybe and then a small message for the students at the end. So first of all, the thing what we have to ask ourselves is, of course, we are having a lot of media thing going on, and also a fact going on that yes, corona is killing over 7 million people. But do we have to really think that is corona the biggest killer in the? And to see this, just I wanted to give a brief idea about how many deaths are happening in the world in a year. Just if you see this chart over here, you understand that over 56 million of the people died in 2017, and out of these 56 million, the infectious diseases account for over 5 million deaths. Whether you take the respiratory infections or the lower respiratory tract infections or tuberculosis. And just I want to mention tuberculosis because tuberculosis is one of the biggest killers all time. It's not that these statistics are not made for 1 year, but yearly and always tuberculosis is one of the greatest killer of all the time. And here, if you see this chart, you can see that tuberculosis kills over 10 million people every year, at least in 2017. And then India has the highest number of the tuberculosis patients when in 2017. So I think we are just ignoring that there are so many deaths happening, but still we think corona is the highest killer of all the time. Yeah. And then, just also understanding it, what are the biggest risk factors of all these diseases, 56 million causes, and then you understand that most of the diseases are kind of a man-made, high blood pressure, smoking, high blood sugar, air pollution, all these things. And I would really want to stress air pollution because, we know the situation, what has happened in India in this year and the last year, a lot of pollution problems. So these are all kind of man-made things. And, these are major concerns through which we can really try to reduce the cost, cost of the lives. And then, coming to the epidemics. So we have the corona epidemic at this point of time, but is it new for us? No. Because you see that there are over 1,307 epidemic events which has happened between the years 2011 and 2017. cholera, Zika virus, and I think all these ones. And I think many of us are also aware of it. And we are already constantly trying to fight about these epidemics. And now we have this new epidemic called corona. And if you just imagine what is the cost of these epidemics and the burden to many countries, the epidemics are not at all cheap, and they are so costly to fight, and they have a big impact on the economies. And over $60 billion is how much the world spends at least every year on these epidemics, what takes place. And then, so I'm now getting close to COVID. And this is the statistics I think I have taken on June 5th. So if you see, we know that United States is the worst affected, and followed by Brazil, and now India is in 7th. And I think India already overtook Italy and Spain as of today is what I expect. And then we are seeing that, okay, is there a flattening in the Europe? Definitely, many people are thinking that there is a flattening in the Europe. And if you see the graph, you can clearly understand, yes, the cases are probably not at least exponential, but still the threat still lies. And then what are the mainly organs affected? First in the beginning, when the coronavirus have started, people said that this is mainly, SARS related, and then it affects the lungs and thereby causing the death of the individuals. But from that point of time, there is a lot of research going on. And then till date, of people don't know like how many organs are affected, what kinds of diseases are affected by corona. And then if you see here, literally COVID is damaging most of the organs. It has an impact on heart, brain, lungs. It's also, I think, recently they also thought that even in the brain it is able to pass, and then it's leading to stroke. All these ones, lungs, intestines, kidney, whatnot. So most of the vital organs are being impacted due to the coronavirus. And then what are the symptoms? These everybody know, but I'll just quickly tell you guys. So the most common symptoms, of course, are fever and then loss of appetite. But scientists have made a lot of emphasis on the loss of smell. And because, this is where the concept of asymptomatic and symptomatic came into existence. And there was a lot of argument in the scientific world that there is nothing called asymptomatic, because even the people who are asymptomatic are not asymptomatic, but rather they do not understand the symptoms. So I think, and the doctors are really telling that these could be identified by the patient so that they can be very early diagnosed. And loss of taste are some of the few symptoms which they said that this could be very much valuable to very early identification of the corona disease. And all others are severe. In the severe stages, you see the difficulty of walking because it might affect the brain, and you get this confusion, and then coughing up of blood and decreased white blood cells, all this. And so I think, so it would be that everybody should be careful in understanding their normal physiology, and even if there is a slight variation, try to understand if there is corona or not. And then what are the other symptoms, which I have recently found out, I think just in last 1 week, it was very heartbreaking that, nowadays they recognize some patients where the placentas of the woman are also affected with the SARS. And then, this leads to the higher rates of decidual arteriopathy, which means, it's a remodeling of the arteries, and this leads to necrosis. And also the maternal vascular malperfusion happens, which means that the blood supply is not properly taken to the kid, and this might lead to several complications such as end organ ischemia as well. And there is a lot of research going on all these different areas. What are the effects of the COVID? And other symptoms also include this purple rashes, swollen legs, clogged catheters, and sudden death. All this, and then large or small. And in terms of the COVID-19, definitely, there are going to be so many challenges, and especially it is going to be for urological communities, what scientists think, and more effects of the organs are being identified on daily basis. And some interesting studies, what I saw about the COVID-19 are that, there's people, now the scientists at least are trying, most of the papers now are just not accepting, publishing in order to produce the data what they have, and most of these have to be still peer reviewed at least. So what they think is like there is an association between the blood group, wherein the blood type A group positive people are have a higher risk of the COVID-19 when compared to that of the O blood group people. And then maybe a little good research, what scientists were also seeing from Germany, what they observed was surfaces could pose only a modest risk for the household spread. Please make this point, household spread, not outside. Outside, you should be always careful, okay? So maybe it's like in the house, you don't need to be extremely cautious, and the spread is not so much, it seems. And recently there is an article, it was also asking, how does Corona-19 kill? And still now, even, there is a lot of research, but people and the doctors and the scientists, we are trying to find out how it is able to kill. There is so much of boom in the research, like a stock market kind of, that in these 3 months alone, there are over 30,000 recent research articles on COVID-19. And then over 5,600 preprints, which is not at all usual. And it is something I have never seen in the research career, that there would be these many publications. And we see the publications, as I already mentioned, that they're not even peer reviewed, but just for the information's sake, they are the journals are publishing on their website. And then, so I would now go to the infection cycle of the COVID-19. So I think this also most of you would have known, but I would just briefly describe you. So first, coronavirus. Okay, this coronavirus is a belongs to the is a coronavirus. Has four. Sorry, I have a small problem with the cursor. Sorry. Yeah. Okay. So this coronavirus has the genome size of 26 to 32 kilobases, and it is one of the largest RNA viruses. Okay, and there are many family members of this coronavirus, and it has four important components. One is a spike glycoprotein, which has two isoforms, S1 and S2. Another one is a nucleocapsid, envelope protein, and the membrane protein. What are the importance of all these? So membrane, if you take the membrane proteins, these are the proteins which are the most abundant. And these play a very important role in the fusion as well as in the later processes of the virus replication. While the spike proteins, these spike glycoproteins are the one where the viruses viruses attached to the host, and later they split into these S1 and S2 subunits. And the S1 subunits forms the head of the spike and has a receptor binding domain called RBD. And this S2 spike, okay, and this S2 anchors the spike in the viral envelope, and on the protein activation enables the fusion. And as I already mentioned, this M determines the shape of also the virus and organizes assembly and interactions. And this E protein.
Hello, Rajkumar sir.
Yeah.
Sir, just a moment, sir. Sorry for the interruption, sir.
Yeah.
Some of the participants
we are unable to see your
to
PPT, sir.
Sorry?
We are unable to see your PPT, sir.
Huh?
Just
Okay.
what you want to do is just stop presenting and repeat the same
Okay.
process, sir. Again, present now entire screen, something like that. Just close, stop presenting.
Okay.
Yeah. Stop presenting.
Mm-hmm.
Yeah. Now once again you share, sir. Present now. Click on present now.
Present now. Entire screen.
Open Google Meet the tab, sir.
Is it okay? Are you able to see now?
No, sir. No.
It's processing.
Yes.
Is it okay now?
Now we are able to see, sir. Just minimize this and open PPT, sir.
Okay. So
Yeah.
I don't
Okay,
know.
sir.
Now we are able to see, sir.
Okay.
Yeah, thank you, sir. Thank you.
So no problem. So I was just mentioning about the different kinds of the important components of the virus, the spike glycoprotein and the nucleocapsid, envelope protein, and the membrane protein. And the membrane protein is the one which determines the shape of the viral envelope and organizes and assembles and the interaction. Okay. And if you're taking this envelope protein is the smallest and plays an important role in assembly and release from the host. And during the replication, it sits on the Golgi complex, and then it plays a role in the entry of the host cell and also intracellular trafficking and all such things. So as I already mentioned, among all these different components, the membrane protein is the one which is highest present, over 300:20 to the envelope, and 1 to the spike protein. This is a ratio in which these different components are present. Okay, and if you're now seeing the virus mechanism, what happens? So first, what happens is the attachment of the virus to the host cell. And once the virus attaches to the host cell, what happens is that the S1 protein gets split. And this S1 protein split leads to the internalization of the virus host membrane fuses of the viral RNA. And after this leads to the translation and cleavage of these polyproteins. And this one later converts into the RNA strand, and then the synthesis of the minus RNA strand and the positive RNA strand. And this leads to the translation of the important proteins of the virus. And these important proteins are similar to that of the host. And that's the reason, so as you all might know, viruses normally use the host machinery in order to replicate. And all these now use up the host machinery and then produce a lot of virus load, and then they mature into the virion, and then they are released. And again, these are now presented to the tissue, and this leads to the activation of the immune system, and later on follows, and then this leads to the disease condition, and later, if it is very severe, this leads to the death of the individual or any serious conditions. And then, as I already mentioned, this ACE2 protein, I think I didn't mention ACE2 protein, the spike protein binds to this ACE2 receptor, which is present on the cells of especially in the lung, is what at least people know. But this ACE2 protein is one of the main components, because without this, the virus cannot bind, is what scientists initially have conveyed. And there was a lot of research, and there are also a lot of drug targets which are targeting the ACE2 so that it prevents the viral entry into the host. But recently, there are some papers which say that this ACE2 is not present in the lungs, or very, very less expressed in the respiratory system, as you can see here. There were many papers in the cell lines, but this is a paper which was published in the Human Tissue Atlas. And then what they found is the ACE2 receptor is mostly present in the gastrointestinal tract, and to an extent in the liver and gallbladder, and also minutely in the endocrine glands, but not in the lung, eye, or other organs. So this was done in almost 150 human tissues and cell types. It was a very proper... Hello?
Oh.
Yeah. Hello. Hello.
Hello, sir. Sir, you can go ahead with your presentation, sir. It is a disturbance from one of the participants.
Okay. Sorry. Okay. So the group has profiled where this ACE2 is present in all these 150 different types of the human tissues and cell type, and they said they have deduced these results that it is not mainly present in the lung. And this is one of the toughest things, that till now, there were some targets, and they really thought that ACE2 is something which can be targeted and then lead to the cure of the corona or something. And then this was the research which has come up. So now I would move on to this COVID-19 drugs. So as I already mentioned you about the ACE2, right? So ACE2 was one of the very early candidates, which was thought to be very effective, and also is currently in the phase 2 clinical trials. And these clinical trials are being carried out by Epiron Biologicals. So what happens? As I already mentioned, the virus first comes and sits on this ACE2, and this ACE2 is cleaved, and this S1 is cleaved, and then this leads to the internalization of the virus particle. It enters into the host and then replicates, etc. So these spike proteins, what they have planned is that the ACE2 recombinant human ACE2 proteins are synthesized so that these go and bind to the virus, And then this leads to the virus unable to bind to the host cell and thereby into the replication, And in this way, they think that the virus cannot infect the humans. And the candidate drug is contaminated. This not only reduces the viral infection, but it also reduces the inflammation. And this APN01 blocks the viral infection by directly binding the viral particles, as well as reducing the disease-mediated organ pathology. But as I already mentioned, if this ACE2 is not present in most of the in the lung or other organs, then we have to find what happens with these clinical trials. And then I think with regarding to the drugs, you all would have heard a lot about hydroxychloroquine and chloroquine. So hydroxychloroquine and quinones are they possess multiple effects, and these are used for malaria drug. This also you are aware of, and they have multiple effects, and the main effects via which these hydroxychloroquine and chloroquine impact are they have these immune modulatory functions. They're also so because of this immune modulatory functions, they suppress the certain immune cell functions, and that is the reason they are used as drugs for autoimmune disease such as lupus, etc. And not only that, hydroxychloroquine changes the pH of the lysosomes, which is used in malaria. And recently, they have also identified that the hydroxychloroquinones act as zinc ionophores. Ionophores are something which pass down the ions in the membranes, allow the passage of the ions. And recently, there is also another paper which says, which have studied that hydroxychloroquinones binds to sialic acids. Sialic acids are a kind of the sugar molecules which are present on the membrane. So there are multiple effects, and all these together, probably everybody think that if there is effectivity of hydroxychloroquine against COVID-19. So what is the mechanism of action? Just in brief. So normally first, as I told, it's an immune modulatory function. It has several kind of in autoimmune disease. That's the reason I'm not showing that one. And change of the pH. So first, once the virus enters into the host, so this is taken into the endosome, and this endosome, only the virus particle is split by the lysosomal enzyme protease, and only then it can be activated. But hydroxychloroquine increases the pH of these lysosomes and then inhibits the lysosomal activity. Thereby, there is no proteases which can cleave the virus, and thereby there is no release of this viral DNA into the host environment, leading to no effect of the virus on the cell. And with regarding to the ionophore property of chloroquine, so normally zinc, if it tries to come onto the membrane and enter into the cell, it doesn't happen. Zinc is bound back. And if there is chloroquine, what happens is the zinc binds to the chloroquine molecule, and then once the HCQ molecule binds the zinc, this enables the zinc to go or enter into the cell. And these metal ions have several impact on the different kinds of enzymes, especially in case of the corona, RNA-dependent RNA polymerase. Without this polymerase, the virus cannot replicate, leading to the effectivity as a drug. And this was recently published in the paper that zinc affects the coronavirus by blocking this zinc ionophore. And the third mechanism of action is on the sialic acid residue. As I already mentioned, the ACE2 proteins do sit on the membranes, right? And the virus molecule comes and sits on these ACE receptors. So recently what the scientists have found is this ACE2 receptor binds, or on the surface, the sialic acid sugar is very close to the ACE2 receptors all the time. And the virus also binds to a little portion of the sialic acid, thereby binds to the ACE2 receptor as well as the sialic acid, and then enters into the cell. So what recently the scientists have just proposed was that if there is if there is an inhibition of the sialic acid, probably there might be a reduction in the entry of the virus load, thereby protective in nature. So yeah. But I think I have explained these three methods, and they sound to be very convincing, right? About hydroxychloroquine on that's data has multi properties and all this. But I think this was also came in newspapers and everything. You should be aware of this. Like, it is so controversial about corona that some companies tell you that, some companies or researchers or countries tell you that no, chloroquine is not effective, or this cannot be used because chloroquine has a property where it leads to reduced heartbeat. And that is the reason some clinicians do not accept for the chloroquine. Whereas some people tell, once there is a disease, immediately, as at a very early stage, if you start the treatment of the chloroquine, then this can be effective. Otherwise, if you're using in the later stages, this has several side effects. And owing to all its useful properties, hydroxychloroquine drug was accepted to undergo the clinical trial, and then few articles proposed that HCQ is associated with the cardiac risk. And what happened, it is just last week, there are two very famous and big journals called The Lancet and also New England Journal of Medicine. These are very reputed journals. And in those journals, some groups in the United States has published that coronavirus do not have any, hydroxychloroquine do not have any effect. Rather, it has a lot of disadvantages and side effects. After publishing within one day, both these very good journals have retracted the paper, which means they said, no, we won't. They published the paper, but then they said, no, this is wrong data. I think the reason was the company has claimed that they have a huge number of samples across the world, that they have tested all these patients who have taken the chloroquine, and then they analyzed the results. So they took it. But then the company did not offer to provide any of these patient data or other things to the journals, telling that this is a privacy policy. And that's the reason these journals have just rejected that. When these two papers have come online, then the WHO, which are conducting the clinical trials on chloroquine, have stopped them, telling that, okay, we have the evidence now, this is not going to be good. And immediately after these two papers were retracted, then again, it's I think 2 days before, again, WHO has accepted and they started the trial again on chloroquine. So, okay, these are mainly the drugs which are of very high focus. And apart from that, there are also some more drugs which are currently under trials, like, antiviral drugs and also anti-inflammatory kind of the drugs. For example, here you see remdesivir, which is the antiviral, because this is adenosine analog, which is very important in the nucleic acid. And without this adenosine, there will not be any RNA synthesis, and thereby the virus cannot multiply, and thereby inhibiting the virus affecting. So this is sponsored by Gilead and WHO, and these are in the phase 3 and phase 4. This is one of the drugs which are in phase 3 and phase 4 stage. And sarilumab, and which is the human monoclonal antibody. So normally it's very easy for you people to understand, like, if there is any drug, I think most of the cancer drugs, all these drugs do have a suffix called MAB, which means monoclonal antibody. And this human monoclonal antibody is targeted against IL-6 receptor and leads to the immune suppression, so that the scientists think that immune suppression can be useful against the COVID. And this is by Sanofi. And similarly, IL-6 has been a good target for many of the scientists, and they have developed, basing on different strategies, the drugs. And the other candidate is tocilizumab, which is a human monoclonal antibody against, again, the same IL-6 receptor. And this is being in the clinical trials via Roche. And the other drugs is lenzilumab, which is the human monoclonal to relieve pneumonia. Some of these drugs are also in the trials for other diseases. For example, for several SARS, Ebola, all these, and they're trying to do this one on COVID-19. And other drug is favipiravir, which is also the antiviral agent against the influenza, which is also currently under testing. And the other one is lopinavir or the ritonavir. So all these are the strategies, antiviral drugs, in short to say. And then we also, I think, we also came up in the newspapers that we have seen some of our indigenous drugs, especially such as ashwagandha, which is the vithanone and vithaferin A, which is one of the compound also from the New Zealand. And these also are predicted to interact with the TMPRSS2, which is the main enzyme involved in the cleaving of the spike proteins, and thereby block the entry of the SARS-CoV into the cells. So this one is in the clinical trial accepted by ICMR and in association with the Japanese National Institute for Advanced Industrial and Science and Technology, along with our DBT. And the other one recently, I think this is also in the last week or something, TCS Innovation Lab in Hyderabad, they also observed the de novo design of the new chemical entities for the SARS-CoV-2 using artificial intelligence. And they identified 31 molecular compounds, which they say they could be very effective when compared to the current drugs which are in the clinical trials. But these are all the models, and they wanted to take to the next level probably. And I think they're offering these models to many other, whoever wanted to perform the clinical trials. And they say mainly that these compounds have more binding properties, and which makes them more effective when compared to that of the other substances which are currently under test. So most of these compounds are the protease inhibitors, which are very much important for cleaving of these virus particles. Thereby, it prevents the replication of the virus. And then coming to the vaccines, because these drugs are all having so many controversies, and also, as I already mentioned, this ACE2 inhibitor, this is not going so far as expected. And the next things, I think, which are very mandatory at this point of time, and the world is waiting for, are the vaccines. And what about the vaccines? So I would just also give you a brief introduction about, I think I'm hearing several news telling that, okay, you will be having vaccine in 6 months, in the next 1 year, or whatever, early stages, but really, at the normal situation, traditional vaccine development frame is it takes at least 10 to 5 years to get a vaccine. And you should also think that there are so many viral diseases till date, and you really don't have any vaccine. There are so few viral vaccines because it is very difficult, and also the virus mutations might be leading to the difficulties in the action against the particular strain. And so in the normal phase, it's you have an idea, and then you try to experiment, and then you try to do it on mice, as we people do it in the laboratories. We try to see the efficacy of the drug in the cell line, and then assume, okay, if it is really working in the cell lines or in the animals, which at least will take 4 to 5 years in the initial phase. And once there are satisfactory results, then it goes to the clinical trials, which at least will take 5 to 7 years. And once all these clinical trials give positive results, these will be submitted to the FDA approval, and then the drug goes to the final large-scale manufacturing. And the cost of the vaccine preparation is also not cheaper. At least it will take up to 500 million to 1 billion from the stage 1 to the end stage, where you get the vaccine into the market. And also now there is a lot of discussion going on. Okay, if you really have the vaccine, can you decrease the incidences of corona? And many of the scientists or the other industries say that this would be still difficult, because already many of the major countries are really giving big bucks for the companies in order to have the vaccine first to them, leading to the kind of scarcity still going on in the developing countries. And also, but this is a case of the very emergency kind of the vaccines, wherein the WHO or the FDA, all these people are really trying to make the permissions as quicker as possible. and also putting in a lot of money, because clinical trials are really, really expensive. And they're trying to put a lot of money, and then trying to see if it is really possible that you get a vaccine at the end of 2020 or maybe 2021 kind of. And then, so what are the different types of the vaccines? Hello? Hello?
Hello, sir. Hello, sir.
Yeah, ma'am, I'm getting some disturbance. Okay.
Sir, we are able to hear you, sir.
Yes. Okay. So there are different types of the vaccines, which are different types of the vaccines in the pipeline for the COVID, and different types of the vaccines include, it could be the RNA vaccines, or they could be the DNA vaccines, recombinant protein vaccines, vector-mediated, inactivated, or live attenuated vaccines. As I have already mentioned, the stages are, first you have this good laboratory practices or medical practices for development, and then they have to go for the clinical trials and phase 1, phase 2, phase 3, where you have this large scale production, and then it has to be administered, and only then they make a very bulk amount of the vaccines ready, and then you get this immunization. So at least as far as I saw in the NIH website or in the clinical trials website, yesterday, at June 2nd, so there are 10 vaccines which are under evaluation, but at least in the advanced stages. But there are over 123 candidates, which they are trying to recruit the people and then trying to start the tests. And then among the vaccines which are in the clinical evaluation, one of the vaccines which is in a very advanced stage is the vaccine from Oxford University, which is being produced from Oxford University Jenner Institute via the group of Professor Gilbert. And this is already in the 2B3 stage, and it was found to be safe for all age groups, and also for those people who are with diabetes, because this is one of the high complications for side effects or other other effects. And then what they did in this vaccine is, for example, they used the chimpanzee adenovirus. This is modified in such a way that this is not harmful. And into this one, they load this CHADOX. This is a kind of a viral vector. This doesn't cause any infection in the humans, and this is packed. And what they want to do is, they wanted, of course, that in this one, they try to sequence the spike protein, and they sequence the spike protein, and then they insert it into these vectors. And then these vectors will be injected to the humans. And what these do is, this artificial kind of sequence will still produce a spike protein. But this spike protein is something which doesn't do anything. And thereby, the spikes are produced, and at a very low dose, the human immune can detect it. And already you have a kind of the protective antibodies against these spike proteins. So whenever the virus comes and tries to attack, then the immune cells already do react to it, and then leads to the immunization. So this seems to be a very effective vaccine. And then we have to see what happens to this vaccine very soon. And then the other vaccine which are in clinical trials is like from the company called Moderna. And this Moderna also uses, also uses mRNA technology. So this mRNA is like, this mRNA, they also isolate from the spike protein of the infected patient, and they try to sequence this one, and then they try to inject this one. So this spike protein is really not infectious, and it is already recognized by the humans, and then it leads to the immune response whenever there is a virus which infects next time. And the name of this one is mRNA 1273. So what are the other vaccines which are in clinical trials? These are mainly from the protein subunit vaccine, which is called the Novavax. And this one is, this Novavax is a very, Novavax. Okay. Novavax is a full-length recombinant SARS-CoV glycoprotein, and this is combined with a nanoparticle. And these nanoparticles then enter into the, and these nanoparticles are injected and leads to the, I think this leads to, it's an adjuvant, and these are injected into the cells of an insect. These insect cells are not harmful, and then these create a kind of a response. I forgot about the Novavax vaccine, and then it creates a response. And then the other RNA vaccine which are in trail is a BioNTech and the Pfizer. And then there are other inactivated vaccines which are being produced in China, 3 of them or 4 of them. And I think India is also producing 3 or 4 vaccines. And yes, these are all live attenuated viruses. So the viruses are completely made as harmless, and then these are injected to the humans so that you get a partly response. The immune system can recognize the virus, and then it creates a kind of a response so that even though if the next time the virus comes, this doesn't affect the individuals. So the candidates are Coragenix, which is from the Serum Institute of India. And this is also in a clinical trial 1 as far as I know now. And then along with the collaboration with the Griffith University, there is this another vaccine by Indian Immunologicals. I did not find the name of this, I think. And also Bharat Biotech from Hyderabad, I think you're aware of it, wherein they're also collaborating with the Thomas Jefferson University, and they also have the recombinant vaccine containing the S1 spike protein. This is a non-replicating viral vector, which non-viral vector, which is also a safer method and thus does not have any effect. And then I think, so with all this, I think, there are a lot of things happening, especially with regarding to the research that there are in certain stages. And then there is research coming up telling that this might not be effective in different ways. And in the newspaper, at least from these big companies like Novartis and Shanta Biotech, these people already tell that it at least take 2 years or 3 years for the vaccines to come. Although currently many of the vaccines are at 3rd or 4th clinical trial phase, because you have to see about the side effects also. Yes, and then, as I already mentioned, you see there are very less vaccines, and also the vaccines for other diseases which are available took almost like 20 years or more than 20 years for getting a good vaccine. Yes, and I think I just want to add that there are new NIH guidelines of how the COVID-19 treatments have to be proceeding. And these guidelines are depending upon the latest research that the COVID is able to infect different organs. And first one is they propose also an antithrombotic therapy. For example, if there is a lot of blood clots, this is what the scientists were telling, that COVID is able to cause some blood clots. And so for the patients which are in the ICU kind of stage, they suggested to use the antithrombotic therapy and the other drugs like remdesivir. For example, this also was initially thought to be very effective, but later they found out some kind of side effects. And then they suggest, as of now, they suggest that only the patients who are hospitalized with severe disease and require oxygen, then they can use this remdesivir as a therapy. And I also already mentioned you about hydroxychloroquine. So they now restrict this to 600 mg maximum, and only if they are if they do not have any issues with regarding to the heart cardiovascular problems. And also they strictly now restrict the use of the HCQ individually. They have to be prescribed, and only then they can be used. Otherwise, they are not allowed to use the HCQ. And the other therapies like convalescent plasma, I think they are tested at some places, but at least the NIH do not propose to use these and also the IL-6 inhibitors, which are thought to be a good target, because the research is coming up which they show that if there is an immune suppression, that might not be good. So this is a problem with some of the agents, like, you do suppress the immune system, but then this have several other complications, or you do get other diseases which might not at all be good for the individual. And then as this came up to me, that I was checking for this, and then the cost of this epidemic is so high, which I have already mentioned. And then, prevention is so better. And this was one of the foundations which have done this study. And they said, like, if at all the governments are able to spend properly and then plan for these epidemics, it would be 60 cents per person, which would be enough in order to face the kind of the pandemics when they do happen. Even at this point of time, why is it so important? I mean, this one is the so-called the best of the countries which can deal the pandemic. If you see, this is the statistics in 2019, and these are the top 10 countries which think that they can face the pandemic. And now we know the fact that United States is the top in the list, where the pandemic has affected very severely, followed by United Kingdom, which was also very badly affected. And then what I think is the need of the hour is, it has to be more funding towards the science and also on the healthcare development. In spite of this, you see, I think this year, in a few months before, the Trump's government have reduced the budget of the science. And this is, again, this is leading to several discussions about this one, like reducing the funds of the science. What does that mean to the health stability across the countries in the world? And then I think we are moving in the right direction, that, with regarding to the scientific temper in the country, that India is able to publish in the top 3 in regarding to the scientific articles. And then it has to be doing more with regarding to the science and research in the country. And I think I also just wanted to add now, like, okay, all this is happening with regarding to the pandemics and the epidemics especially. And I would like to ask, how responsible we are, and then how are we contributing at this time of epidemics and the pandemics, and for especially for our future, And I think there are plenty of problems, at least dealing with the pandemics, the way how we behave, and then how we are spreading the disease among ourselves. And then, it has to be that we have to think the solutions together. And I think especially in the colleges or in the educational institutes, these have high responsibility in order to follow all this. And also the faculties should be supporting these young minds towards these innovative ideas, which has to be promoted. I think this is possible only once there is a good amount of the research also going from the level of the schooling, so that would be inspire the young minds for the future on. And then, just why I meant this one is I was just thinking, how are the colleges, everybody going to react at this pandemics when there is nothing to do, or what are the projects? Of course, you have some online classes, but beyond this, if there is no vaccine, are you going to be 2 years at home, or what are you going to do? So maybe I think this is a good time that you find solutions, how the colleges has to be in the regular kind of mode to be placed on. And then, and just not these pandemics. We have a lot of problems with the pollution, food habits, as I already mentioned in the first stage itself, food habits, especially diabetes and the smoking, which are leading causes of the death if you combine, and also the cancers. All these are the basic things which could be done and which has to be fostered since the young age. And I think this is a good time for everybody to think of it. And then just come up with your ideas, and then try to excel in your careers is what I would like to tell. So in conclusion, what I wanted to tell is, yes, with regarding to the pandemic, there is Europe which is flattening, but I think many of the scientists across the countries are suggesting that there could be a second wave of the disease, which is really expected. And after all my talk, I think you would have realized that it is not so easy to make the vaccine, and this is not going to be a simple task in the near future, at least in the next 1.5 to 2 years. And of course, but maybe many vaccines are at advanced trials, and there are a lot of collaborations going on in order to really test large number of the patient samples so that you get enough amount of the data, which is the main criteria in order to proceed on to the next level and for the mass production. And I would just think that, creative or innovative ways have to be provided, have to be thought of in order to prevent it, so that at least break the chain will be happening, which will definitely reduce, because we have seen many countries, especially I would also tell Germany and other countries, wherein the break the chain have really worked out, and staying at home really decreased a lot of cases. Yeah, and yes, prevention is better than the cure, and I would wish that people will be staying healthy and with all this. And with this, I would like to acknowledge the principal of Stella College, Dr. Jacinta Quadras, and also the head of the department, Lasina ma'am, and all the team members, and also my teachers and my family members and people over here from Pharma Centrum in Frankfurt, which are being of great support to me. And I would also like to thank all the people who are doing a great job back there in India. And then please stay home, and then try to be protective. So with this, I would like to thank you, and then thanks for your attention, and I would be happy to take your questions. Yeah. Hello. Hello.
Hello, sir.
Yeah.
Hello, sir. We have a question from Dr. Mannava Madhavi, ma'am. She's asking, what is the lifespan of antibodies to coronavirus in convalescent persons?
Actually, it depends. It depends upon the vaccine type to vaccine type, and also vaccine to vaccine also. So these are all in the clinical trial data information, and I think this has to be checked. But it depends upon which vaccine you're talking about.
Hello, Raji.
Yes,
Rajeshwari.
Yeah, sorry.
Hello, Rajeshwari, ma'am. Are you able to hear me? Okay, sir. Sir, we have one more question from Samina Banu. She's asking that how can we overcome this anxiety or unpredictable situations during pandemic?
I think the anxiety and these things come only when you have nothing to do. I think just try to involve in some kind of the work. You have so many things to do, and I think definitely you will not be getting any such thought.
Okay,
because
sir,
this delirium, and many of the people have this effect, and as I already mentioned, brain disease is delirium. All these things are really stressed out, and then psychological behavior is really changing. And there are many papers which are coming up with this also. But I think it's a simple solution. At least in India, we have a lot of people together. So just spend time and then read. There are a lot of things to study.
Okay, sir, we have one more question from Siddhi Jani. How vaccine will work because it is keep on mutating?
Yes, exactly. So yes, definitely that is the one of the challenges of the vaccine, and that is the reason people are really careful in order to design. For example, sometimes if it is a spike protein, this also might change. But this depends. Yes, this is a challenging part of producing the vaccines for the viruses, especially, and that is the reason you cannot see a lot of vaccines for the virus. I heard that, I think, at least as far as my knowledge is concerned, there are over 200 mutations already identified, and 2 or 3 types of strains are already there with this COVID-19. But still, I think spike proteins, these are not something which change so aggressively, is what I suppose. And that's the reason there are other, for example, there are many clinical trials which are also taking place with combination therapies sometimes, if it works. For example, HCQ, it has multiple effects, right? It's a drug, but it still has a multiple effects. So and also the clinical trials are going on regarding to the combination of 2 drugs also, so that it could be having a desired effect. But vaccine-wise, I think most of these are not so sensitive, or there are some conserved areas. If these conserved areas, the vaccines are developed, I think then it would be then it would not be such mutating kind of. So I think the strategies are like that in such a way.
Thank you. Sir, we have two more questions. May I ask, sir?
Yes, ma'am. Yes, ma'am.
Sir, we have a question from Junior College BSSP. They are asking, sir, are there any rapid tests to find the COVID-19 other than swab
Yes.
test?
Yes, for example, swab test. Apart from that, as I already told you, right, many of the if you really want to confirm this one, I think in India also recently they have developed one test called Feluda, which would be probably coming into a market in 2 to 3 months. And I think this would be giving the result within half an hour or something. This is developed by NCCS or some institute in Kolkata. They have developed this, and this is really a very effective kind of a diagnosis, and I think this will be soon coming up.
Thank you, sir. Thank you, sir.
Yeah.
Thank you, sir.
Yeah.
Sir, we have a question from Presentia Aloysius. If the malarial drug HCQ is yes, HCQ is good, are there any common factors between COVID-19 and malarial parasite? They're asking, sir.
Malarial parasite? No, it is mainly, it depends. The main mode of action of this could be completely different with regarding to these two. But the common thing which HCQ does is that HCQ, I already told you, that increases the pH of the lysosomes, right? This is also very important in case of the malarial parasite, which leads to the cure of the malaria. So in this aspect, yes, the action of the HCQ is similar, but not about the parasite itself. But the mode of action of HCQ is common in both of the things. It just increases the pH of lysosomes.
Okay, participants, any more questions? Dear participants, any more questions? Okay, it's wonderful. Now, it's time for the end of this session. Thank you all. Now it is time for vote of thanks. It's really been a fascinating session. First of all, I wholeheartedly thank Dr. Rajkumar Uttaguri sir for spending your precious time with us and generously contributing the information about this pipelines of COVID-19 vaccines and their trials. Sir, you led this webinar to the higher level with your eloquent presentation, sir. Really, it's a pleasure to have you among us today as a resource person. Have been exceptionally helpful to organize this webinar in such a great extent, sir. Thank you so much. And I also thank all the participants from nook and corner of India, and also some of the participants from Macquarie University, Sydney, Australia, and the Royal University from Melbourne, Australia, and Kingston University, England, for your overwhelming responses and your kind cooperation with us. I express my heartfelt gratitude to our beloved principal, Dr. Sister Jacinta Cortez, for her constant support and encouragement in all the ways and means to organize this international webinar in such a great extent. Our IT member, Mr. K. Raju, for his huge technical support to organize this webinar, and my department member and co-convener, T. Rajeshwari, for her support and help. And I thank all of you for this webinar. Dear participants, we will send the feedback link to the WhatsApp group as well as to your respective mails. You will get the certificates within 1 week. Thank you, one and all.
Thank you so much, sir, from Stella College, and thank you so much.
Okay, okay. So now I could log out, or what should I do now?
You can
Or
directly close, sir. You can close the tab.
close it.
Yeah.
Okay.
To left the yeah.
Okay.